So now we're using FRAP. We're going to analyze FRAP. But acetate buffer 0.3 molar. We're going to use the same plate. So to prepare the FRAP, 10 milliliter of the acetate buffer. And 1000 microliter of ferrum chloride, 20 millimolar. Vortex first. And this is TPTZ, 10 micromolar. Vortex first, and then 1000 microliter. Into the FRAP solution. This is the color it should have. You can see it. Vortex. What a nice plate. Look at the colors. So 20 microliter from the samples and calibration curve into the plate in 3 replicates. Next concentration of the calibration curve, 3 replicates. Third concentration. 100 microliter of the FRAP solution goes into each one. Already see the differences in colors. These are the low concentrations, these are the high concentration, these are the samples, and you can already see that the samples are too dark for the calibration curve. So the concentrations here are too high, and they're higher than the highest concentration of the calibration curve. So we're going to dilute the samples in order for them to fit into the calibration curve. You can see it also. You can see it's already too high. So that's 80% ethanol for diluting the samples. So that's 1,000 microliter, and we're going to take 1,000 microliter from the ethanol 80%, and then 100 microliter from each sample in order to dilute. Into 1,000 ethanol. This is one sample, and then the other sample. Diluting 100 microliter into 1,000 microliter, 80% ethanol. And now we're going to do the readings of the diluted samples. Next, you can see the color is lighter now, not as concentrated, of course. Color of the second sample. So again, 20 microliter from the diluted samples, as we did before, just with the diluted ones. This is sample number 1. This is sample number 2, diluted. אוי, זה לא מצלם. לא, זה מצלם, זה מצלם. And 200 microliter of the FRAP solution. Solution into the diluted samples. You can now see the difference in color. It's much lighter. It's somewhere around here in comparison to the calibration curve, so it's good because it's in the middle of the calibration curve. then we're gonna let it stand and then take it into the spectrophotometer, to the plate reader, and read it. You can see the differences between the undiluted samples here. Here, undiluted samples, these two, and here the diluted samples. And you can see that the colors are just inside the calibration curve.